Okay, folks, excuse me, let's get started. So as you can see, I've got grades posted for the exam. It was a very interesting exam, grade-wise. Average 75.2. I think combined with the high average that you guys had last year, this class actually has 2 of the 3 highest averages I've ever had. So more to your credit. That's very good. The low was about 26. The high was 100. I think there were 4 people who got 100. So that was very impressive. I was quite impressed with what I saw looking through things. I thought that you really did a very good job on this. One of the things that probably was a factor on this exam was because of the rain day. We had a little bit less material on this exam, and so consequently on the next exam we're going to have a little bit more material. So if you guys do this again on the next exam, I will be delighted. I really will. So that would be very cool. So keep up the good work. If you're finding that you're having difficulties and you need assistance or a tutor or anything, please let me know. I have some very good students who are tutoring, and I can connect you with them if that would be of assistance to you. Or if I can be of assistance to you, of course, let me know as well. Okay. I'm going to finish up, and we're actually going to get caught up and might even get a little bit ahead today with respect to fatty acid metabolism. And that will lead us into prostaglandin synthesis and metabolism. And then I may actually get into talking a little bit about nucleotide metabolism if I finish early today, which I think I will. You've seen how fatty acid oxidation occurs, and you saw how fatty acid oxidation actually related to the synthesis of ketone bodies. If you recall, the synthesis of ketone bodies was the reverse of the very last step of beta oxidation, because thiolase was putting together 2 acetyl-CoAs to make that acetyl-CoA and ultimately ketone bodies that were useful for keeping the brain alive and the rest of us alive as we're very low on glucose. Fatty acid synthesis, as I've noted before, and I'll go through it again with you here today, also is very closely related to fatty acid oxidation in the sense that it is chemically, very simply, very much like the reverse of that of oxidation with a few differences, as I have noted. So I want to spend just a little bit of time summarizing those reactions one more time for you, and then I will talk about the regulation of fatty acid biosynthesis, which is interesting and important. Okay, so looking at fatty acid synthesis, that's not what I wanted. We basically saw, that's not what I wanted either. There we go. We basically saw a chemical reversal of the oxidation reactions, starting with a condensation, whereas before in the corresponding reactions in the oxidation we had thiolytic cleavage. A reduction, where previously we had an oxidation. A dehydration, where we previously had a hydration. And last, a reduction, where previously we had an oxidation. Now, you notice on this scheme some of the differences compared to fatty acid oxidation. One is that the electron carrier is NADPH. And I haven't mentioned it before, I don't think. If I have, I'll repeat myself then. And that is the NADPH is commonly used as an electron carrier for biosynthetic reactions. Very commonly a carrier for biosynthetic reactions. It's not absolute for biosynthesis. For example, you saw NADP being used in the, NADPH being used in the oxidation of unsaturated fatty acids. But in general, NADPH is used for biosynthetic reactions much more than NADH itself is. Okay. Well, the product of this reaction, as I noted, gave us, in this case, a 4-carbon intermediate. And that 4-carbon intermediate becomes then, in essence, this acetyl-ACP, except for now it has 4 carbons instead of 2 carbons. Okay. You'll notice then that the incoming new carbons come with this malonyl ACP. And so the previous ACP gets displaced, and what it was attached to gets attached right there. Okay? So the newest carbons will always be on the end linked to ACP. And that means that the ACP itself will always be the newest one on the molecule as well. Okay? Let's see, what else am I going to say here? So I remind you that fatty acid biosynthesis occurs in the cytoplasm, not in the mitochondrion. And it turns out that the cells have to get acetyl-CoA out into the cytoplasm. I'm going to talk a little bit about that relative to respiratory control in just a little bit. But suffice it to say that there's a very useful and simple mechanism that cells have for getting that acetyl-CoA out into the cytoplasm where the synthesis of fatty acids occurs. And that actually is my good lead-in for this right here. Well, before I say that, fatty acid synthase, again, I'm not expecting you to know this figure, so don't sweat it. The fatty acid synthase, you recall, is the complex that makes the fatty acids. It has multiple catalytic activities on it. These are schematically shown by these various letters, or two-letter scheme that's up here. And literally, this fatty acid, as it's being synthesized, goes around the clock. Each time, adding 2 carbons, getting reduced, getting dehydrated, etc., all the way through until finally we end up with a fatty acid that is 16 carbons long. So the final product of fatty acid synthase is a 16-carbon fatty acid known as palmitate. Okay, it's a 16-carbon fatty acid known as palmitate. Well, that isn't the complete end of the story, because palmitate, first of all, has 16 carbons. It is full of saturated bonds. There's no unsaturated bonds in palmitate. So we have to account for 2 things. We have to account for the longer fatty acids, and we also have to account for how unsaturation gets into the molecules. Okay? So let me account for those before I come back and talk about how we get acetyl-CoA out there. All right, so I don't have any great figures for this, unfortunately, but it turns out that these are relatively simple. All right? The first that I will talk about is the unsaturation or the desaturation of fatty acids. This is the act of incorporating double bonds into the fatty acids as they're being synthesized. And there is a fairly elaborate scheme that we used to teach. I'm not going to go through that scheme, and we're going to keep it very simple with you. There's an enzyme, the class of enzymes that catalyze the formation of double bonds in fatty acids are known as desaturases. And desaturases are found in the endoplasmic reticulum. So that means the fatty acid will have to get to the endoplasmic reticulum in order to have double bonds put into it. The desaturases put in cis bonds only. That's one thing about the desaturases. They put in cis bonds only. And if you're an animal, it puts in cis bonds only up to position delta 9. Okay? So delta numbers from the carboxyl carbon towards the methyl. If you start with carboxyl as number 1, okay, the desaturases in animals will put it in carbon number 9, but it will not go further than carbon number 9. As a consequence of that, and we need fatty acids with double bonds beyond carbon number 9, it means that we can't make the fatty acids that have multiple double bonds or have bonds beyond carbon number 9. Those fatty acids are what we call essential, meaning that they have to be in our diet. And of the ones that you see on the screen here, these would include, okay, so here's, these are omega designations. So omega designations are not good for our purposes because they're counting from the methyl, and the methyl will depend on how long the carbon is, so we can't really count there and get what we want. But of the ones on the screen, palmitoleic is a non-essential fatty acid. Oleic acid is a non-essential fatty acid. But the 2 above it are both essential fatty acids. Okay? I'm sparing you some structures here. But suffice it to say that both linolenic and linoleic are both polyunsaturated. Linoleic has 2 double bonds. Linolenate has 3 double bonds. Okay? All right. So, yes? Okay, so linolate has the omega-6 here. It has 2 double bonds. Linolenate has 3. Yes, Lynn?
Do you talk about animals that mean other like the mammals and reptile fish, or just mammals?
Yeah, that's a good question. I said animals, and actually what I really mean is mammals. Mammals are the ones that are that have this limitation. So replace what I said animals by mammals. Yeah. Okay. So for mammals, we've got to have these fatty acids in our diet. They're abundant in plant tissue. And so one of the reasons we should get some good plant consumption in our diet is to get essential fatty acids that are in there. Or we get them out of animals that we eat that have eaten it themselves from a plant, etc. So those are also important. Okay, so desaturation is fairly straightforward. There's also something I want to talk about, and it's not even, your book used to talk about it in the old scheme, and it's not even there in the new scheme, and that's elongation. Okay? So elongation of fatty acids is also important for us to recognize, and elongation turns out to be kind of interesting. Okay? So elongation occurs as a result of action of enzymes known as, and hold on to your seats for this name, elongases. Okay? Kind of a surprise, huh? And elongases catalyze the very same types of reactions that you've already seen for the general fatty acid synthesis. Okay? So they'll go through that condensation. They'll go through that reduction. They'll go through that dehydration, go through the reduction again. All right? These desaturases are located in 2 places. And the second place will surprise you. The first place we see the desaturases is in the endoplasmic reticulum. Yes, Anisia? Oh, elongase. I'm sorry. Yes, sorry. Your brain latches onto a word, you know, and it won't let go of that word. Elongases, yes. The 2 places we find the elongases are in endoplasmic reticulum, and we also find them in the mitochondrial matrix. What the heck is that all about? All right. I thought you said, Kevin, that the mitochondrial matrix was where fatty acid oxidation occurs. Okay? And I said, yeah, that's right. Fatty acid oxidation occurs there. And we've got elongases there. So how does that work? I'll ask you. How does that work? That's a nice thought. Do they also catalyze the reverse reaction, that is to help break it down? Well, really, no. They're making fatty acids in the mitochondrial matrix. Anesia? Well, they do build into things your body needs, definitely, that's for sure. But there's a reason why this is possible. Jared? Not because of energy use. Not to do with the odd number of carbons. No, keep going. Is it for the mitochondrial membrane? There may be some truth to that. There may be that's why, but I'm sort of asking you how. How come this doesn't get all mucked up? We've got synthesis and we've got oxidation going on in the same place. Jody?
Can the oxidative enzymes not attack something that large?
Ah! Jody says, can the oxidative enzymes not attack something that large? You actually know the answer to that question, don't you? Okay. Where did we have the first enzymes involved in oxidizing long fatty acids? Were they in the mitochondrial matrix? Nope. They were in the peroxisome, right? So it turns out that once you get these big fatty acids in there, all right, the mitochondrion can't do anything with them. So what does it do? It makes bigger ones. It's an odd scheme, but that's what happens in our cells. So knowing where the acetyl-CoA dehydrogenases are located allows us really to answer this question. You can see that there's no enzyme there that'll start that oxidation process in the mitochondrial matrix on a fatty acid that's 16 or longer. Okay? Yes, sir?
And then what's the mechanism for transporting something that large back out to be used?
What's the mechanism for transporting something that large out to be used? It's a good question. I don't have an answer for it. And they do obviously have to be transported out. I don't know exactly how that's done. And keep in mind that they're made in two places. So the person who said that they're made and used in the mitochondrion, probably that's what why the mitochondrion is making some of its own, is for itself and doesn't have to move them back out. The endoplasmic reticulum, on the other hand, does have to have the mechanism. And it's probably the same mechanism that the endoplasmic reticulum is using to export the unsaturated ones as well. So probably both are handled by the same export system. But I don't know what the export system is. Okay, good. Good thoughts. All right, so that's what's happening with fatty acid synthesis for long fatty acids and for unsaturated fatty acids. So we look at those in a little simpler way than we did before. Okay, well, I mentioned that we had to get acetyl-CoA out into the cytoplasm where synthesis occurs, and this schematic shows us how that process occurs. So it turns out that acetyl-CoA itself cannot be moved across the intermitochondrial membrane. Instead, the cell uses the molecule citrate as a shuttle, that is, to carry the two-carbon piece across. So here's the mitochondrial matrix on the left. In the mitochondrial matrix, we have the reactions of the citric acid cycle up to citrate. Citrate can then be moved with an antiport that will move citrate out, and at the same time it moves pyruvate in. Okay? Now that turns out to be important for our purposes for metabolic control. All right? Citrate gets out in the cytoplasm, and it gets broken down into oxaloacetate and acetyl-CoA. Acetyl-CoA can then be joined to acyl carrier protein, and fatty acid synthesis ensues. Jody.
Does it use any of the same enzymes that are in the citric acid cycle, like citrate synthase right there in reverse?
Good question. Is it using same enzymes of the citric acid cycle? And the answer is no, it's not. Because the citric acid cycle enzymes are specific to the citric acid cycle. The enzyme it takes to break this guy here is called citrate lyase. And that sounds like that glyoxylate enzyme that I talked about earlier, which was isocitrate lyase. Okay? It's not the same enzyme, obviously. And citrate lyase cleaves citrate into acetyl-CoA and oxaloacetate. So there's a special enzyme out in the cytoplasm that does that. Now, I want to spend a minute talking about what you see on the screen here, because it really relates again to this business of energy and obesity. Okay? Very important for us to understand what's happening here. Well, when is this process going to be kicking into place? Well, the most obvious time when we will see this process kicking into place is when citrate concentrations start increasing. Citrate concentrations start increasing. Well, how will citrate concentrations be increasing? Okay? Well, let's imagine the following scenario. Let's imagine that I am eating more than I'm burning. Let's imagine that I'm eating or drinking high fructose corn syrup that we talked about last term. If we talk about and think about what happens with high fructose corn syrup, I said that what happens is we bypass the PFK reaction in glycolysis, and we start forcing production of pyruvate. This is Kevin Ahern's pet theory about why people are becoming obese. We're coming back to it again. All right? High fructose corn syrup is forcing production of pyruvate that non-high fructose corn syrup will not, at least to the same extent. Okay? It means we have an abundance of pyruvate. An abundance of pyruvate coming in comes in right here. It doesn't have to go through this circle as you see it here. If I got a lot of pyruvate floating around out here in the cell from glycolysis, bang, it's in. Okay? If I'm eating and or drinking more sugar than I am burning, what's going to happen? My energy state going to be high or is it going to be low? High. I have an abundance of ATP, right? Because I'm not exercising enough. My ATP levels are high. I'm not burning that ATP off. What happens to my ADP levels? Low, right? ADP levels are low. What happens to the oxidative phosphorylation? It slows down. There's no ADP to convert to ATP. What happens to proton gradient? It goes up, right? Proton gradient goes up because the protons aren't coming back in. Proton gradient goes up. What happens to concentration of NAD? It goes down, right? Because there's no place to put the electrons. The electron transport is stopping, right? If NAD concentration goes down, what happens to the citric acid cycle? It stops. And where are those electrons needed in the citric acid cycle? Oh boy, now you're going to stretch your memory. I'll tell you. The first place they're needed is isocitrate dehydrogenase, after citrate. So what's going to accumulate when you're doing all these things? Citrate. And when citrate accumulates, what happens to it? It goes out in the cytoplasm. And what happens in the cytoplasm? We make acetyl-CoA and we make fatty acids. Now Kevin Ahern's pet theory about why Americans are becoming obese is complete. Quit eating all this crap. I say this as one who does that. All right? So that's what's happening here. If we force production of pyruvate, we're going to force this process, and the results of forcing this process are to move acetyl-CoA out into the cytoplasm with nothing to do but be made into fatty acids. And guess what? Remember, our ATP concentrations are high. And what does it take to make fatty acids? All this stuff that we've been talking about. All this stuff that we've been talking about. Bang. We're in trouble. We break the cycle by breaking the amount of pyruvate that we start feeding into the system. We reduce our intake of sugars. People scoff at low-carb diets. But in fact, low-carb diets are the easiest way for you to reduce your pyruvate concentration. That's the way to do it. That's the way to reduce it. Okay, so that's what's happening with obesity, or one of the ways that we can become obese. Fatty acids, of course, lead to fat. All right. The last thing I want to talk about with respect to fatty acid metabolism is to talk about regulation. Okay? And I briefly mentioned it before, and now I'll talk about it in a little bit more depth. Okay? So, acetyl-CoA carboxylase is the only enzyme in fatty acid biosynthesis that's regulated. Okay? That's the very first enzyme, and that was the enzyme that put that carbon dioxide onto acetyl-CoA and made malonyl-CoA. That's a necessary step in making fatty acids. That enzyme is regulated in our cells. That enzyme is found, of course, in the cytoplasm, where all the enzymes of fatty acid synthesis, general fatty acid synthesis, are found. Okay? And we see one regulatory scheme up here. The enzyme is partly regulated by phosphorylation, dephosphorylation. Okay? This is an AMP-activated protein kinase. An AMP-activated protein kinase. Why would an AMP-activated protein kinase be useful here? What does AMP tell us in the cell? Low energy state, right? Low energy. Do we want to be wasting energy on synthesizing fatty acids? No, we don't. So we phosphorylate the carboxylase to at least partly inactivate it. Okay? If we want to activate it, we need a phosphatase, and phosphatase brings it this way. How do we activate phosphatases in the cell based on what we learned last term? Insulin. Okay? Insulin. Insulin is going to favor activation of acetyl-CoA carboxylase. Insulin is going to favor the synthesis of fatty acids. And when do we make plenty of insulin? After a big carbohydrate consumption. So again, all the wheels are turning in favor of making fatty acids the more carbs we take. The more carbs we take, the more we're favoring insulin, and the more we're favoring the activation of acetyl-CoA carboxylase. Now, I said that phosphorylation is one way of partly inactivating it. There's another way of partly inactivating it, and that is by the byproduct of fatty acid synthesis, palmitate. Palmitate will inhibit acetyl-CoA carboxylase. Oh, good, we're in the good, right? Well, maybe not. We're also in the bad. Because there's also another allosteric effector that activates the enzyme. And that allosteric effector is citrate. Where do we get citrate? Well, you saw it in the last scheme. It's coming out from the mitochondrion. As citrate starts accumulating, what's going to happen? We're going to activate this enzyme. Now, we have different degrees of activation. We see the most inactive enzyme is going to be phosphorylated with plenty of palmitate around. The most active form of the enzyme is going to be dephosphorylated with plenty of citrate around. And somewhere in between with the other two states. Now, this enzyme is also of interest in another perspective, in that the enzyme exists in the cells as long polymers when it's dephosphorylated. So the active state of the enzyme is a long polymer. You can actually see it in a microscope. Okay? Really long polymers that go one to the other, to the other, to the other, very active in the polymer state. Putting the phosphates onto it causes the polymer to fall apart. And it becomes monomers at that point. The monomers can be somewhat active, but not nearly as active as the polymer. Here are the filaments right here. Those are actually acetyl-CoA carboxylases that you can see. Okay? Pretty amazing stuff. There's a lot of enzyme to make polymers like that. Okay. And phosphorylated, dephosphorylated, that's just demonstrating to you graphically what I told you in words. That is, the dephosphorylated, the most active, highly phosphorylated, the least active, and citrate concentration affecting those activities. Okay. Let's see. I think that's basically what I want to say about fatty acid oxidation. I won't go through that. Questions or comments before I talk about prostaglandins? Should we sum it up in a song? Okay. So I've got a song, and by now you guys should know this tune because it's the third time in a row we're using the same tune. I can promise this is the last time we'll use this tune this term. It's When Johnny Comes Marching Home. Okay, so the 16-carbon fatty acid palmitate gets all the carbon that it needs from acetate, which citric acid helps release from mitochondria matrices. Oh, a shuttle's great when acids are synthesized. Carboxylase takes substrate and it puts within dioxycarbon carried on a biotin. CoAs all gain a quick release, replaced by larger ACPs, and it all begins when acids are synthesized. A malonate contributes to the growing chain. 2 carbons 7 times around again. For saturated acylates, there's lots of NADPH that you must obtain when acids are synthesized. Palmitic acid made this way all gets released. Desaturases act to make omega-3s. The finished products, big and small, form esters with the glycerol. So you get obese when acids are synthesized. That's the bad news, right? Okay. What's that? Bad news at the end, yeah. Okay, so the last topic I'll talk about relative to fatty acid biosynthesis is that of the synthesis of arachidonic acid and prostaglandins. And these are really interesting and odd compounds. Okay? They're interesting in that they cause many different phenomena, or are linked to many different phenomena in the body, and they do arise from fatty acid biosynthesis. So let's take a brief look at what happens with these guys. Okay, so I haven't talked about arachidonate. I need to do that. Arachidonate is a 20-carbon fatty acid. It's a 20-carbon fatty acid. It has 4 double bonds in it. It's very unsaturated. Okay? Normally, we don't keep arachidonate floating around in our cells freely. And the reason that we don't is it's very, very readily converted into leukotrienes going above or prostaglandins going below. These guys actually cause some similar things to happen. The simplest and most common things that we associate with prostaglandins is pain. Okay? Well, if we don't have arachidonate floating around free in the cell, where does it come from? Right? Well, as soon as the cell gets some arachidonate, what it does is it bundles it up into glycerophospholipids. Okay? It doesn't want it floating around free, so it puts it onto a glycerophospholipid, and you know where glycerophospholipids end up. They end up in your cell's membranes. So those arachidonates are sitting there waiting to be released. Okay? They can come either directly from phospholipids, which are the glycerophospholipids I'm talking about, or if we had diacylglycerol, how would we have diacylglycerol in our membrane? Anybody remember? Anybody remember IP3? PIP2, IP3? All right. So when we clipped off that inositol, that inositol phosphate, we're left with a diacylglycerol in the membrane. Okay. So in either case, we've got something that's got two fatty acids on it, and one of those fatty acids in our scheme here has arachidonic acid on it. Okay. So that can be clipped off. The arachidonic acid can be clipped off by either a lipase cutting from one side, that is by cutting diacylglycerols, we need a lipase, or if it's cutting off of a glycerophospholipid, it's something called phospholipase A2. You can call it PLA2 if you want. Okay. Now, I'll come back to PLA2 in a minute because it turns out to be one of the ways that we can control the synthesis of prostaglandins. Okay? Well, once we have arachidonate, I'm going to focus our attention going down into prostaglandins. All right? Going down. All right? There's an enzyme that converts arachidonate into prostaglandin H2. And I'll show you a structure very briefly, just to give you an idea about what kinds of compounds we're talking about. Prostaglandins generally look like this. If you count, they'll have 20 carbons. Okay? They'll have 20 carbons. And they will always have a sort of a ring structure at the end, like this guy. Okay? So there's only one prostaglandin here. There's actually many prostaglandins. But they all have a general structure that looks like what you see here. Okay? This is what I call a cyclic structure, this little ring that's here. If we compare them to leukotrienes, we don't see that ring. So this is a linear structure. This is what I call a cyclic structure. Okay? All right. So all prostaglandins will have that cyclic structure in them. All right. Now, they also gain some oxygens. Remember, we're starting out with fatty acids. We started out with a 20-carbon fatty acid, and you haven't seen fatty acids that have oxygens in them. That meant that something had to put those oxygens in there and make that ring. So now let me come back to the scheme and show you what happens. We go back to the scheme that I showed you before. Prostaglandin synthase is the enzyme that does both of those things. Prostaglandin synthase isn't usually called that. It's usually called cyclooxygenase. CYCLOOXYGENASE. A cyclooxygenase, again, now the name tells you what it does. It makes that cyclic ring, and it puts oxygens on there. Cyclooxygenase. People frequently abbreviate cyclooxygenase as COX, and they call this a COX enzyme. Okay, well, why do I tell you that? COX enzymes turn out to be of incredible interest scientifically. Okay? And they're of incredible interest because we have a lot of interest in inhibiting the action of cyclooxygenases under certain conditions. So they're very much a target for drugs to inhibit the action of the enzyme. Why? Well, there's many times we want to inhibit the production of prostaglandins. You have a headache? you're producing prostaglandins. What do you take for a headache? You take a prostaglandin inhibitor. Aspirin is a prostaglandin synthase or a cyclooxygenase inhibitor. It inhibits the production of prostaglandins. Okay? Ibuprofen is, in fact, a prostaglandin synthase inhibitor or a cyclooxygenase inhibitor. Most people commonly say it's a COX inhibitor because that's what it is. Okay? Now, aspirin, in particular, inhibits the enzyme by making a covalent bond with the enzyme. Okay? It makes a covalent bond with the enzyme. That stops the enzyme from functioning right in its tracks. Okay? Now, aspirin is an example, and ibuprofen, any of the inhibitors that inhibit this enzyme, this COX enzyme, any of the inhibitors of this COX enzyme are known as non-steroidal anti-inflammatory drugs. Okay? NSAIDs, N-S-A-I-D-S. You've heard of NSAIDs before. NSAIDs inhibit this enzyme. Non-steroidal anti-inflammatory drugs. That name tells you, oh, I bet prostaglandins are involved in the inflammation response as well. And the answer is, yes, they are. If you get a bee sting and it's hurting and it's swelling up, the doctor says, take some aspirin. It will relieve the pain. It will also relieve the inflammation that happens as a result of that. You have knee joint problems or something, the doctor's going to say, take some aspirin, and will reduce the inflammation and hopefully reduce the pain in the process. Prostaglandins are involved in both of those processes. Okay? All right. Well, why do we say that they're non-steroidal anti-inflammatory drugs? If we say there's non-steroidal anti-inflammatory drugs, that must mean there are some steroidal anti-inflammatory drugs. And the answer is, yes, there are. They inhibit phospholipase A2. Okay? Corticosteroids are commonly used to inhibit PLA2. Now, while these guys give some reasonable relief, the steroidal anti-inflammatories give great relief. Okay? So I always tell the story, when I first came to Oregon many years ago, before all of you were all born, I decided on my very first week here, I'm going to go out, and there's blackberries everywhere, and I've never been any place where blackberries grow wild, so I'm going to go out and pick blackberries in my shorts. Oh, I was so young and foolish. And I got this biggest batch of blackberries that you ever see, and then I got to sort of, what the hell is that, you know? Well, I got into poison oak like you'd never seen, okay? And I literally had my leg twice the size of normal. I mean, I was a little scared, shall we say, plus in extreme discomfort. Went to the doctor, and the doctor looks at that and he goes, whoa, we're doing something with that, and put me on steroidal anti-inflammatories, stopping as much as possible the synthesis of arachidonic acid. This pathway coming from right into here is very minor. The vast majority of arachidonic comes from this pathway, and by stopping the production of arachidonic acid completely, the swelling was brought down. So when you have people who have a big problem with inflammation, big problem with swelling, they'll usually get put on to a steroid to help reduce that. Okay? Because the steroids are very effective at working right here. Steroids are not good in general to take, so most of the time we try to inhibit this guy, but in a severe case, we'll use a steroid to inhibit that. Okay. Now, prostaglandins, I will tell you there are many prostaglandins. Some prostaglandins involved in pain, some prostaglandins involved in inflammation, some prostaglandins are involved in uterine contraction during the birth process. Okay? Some prostaglandins are involved in making your blood platelets sticky. They help the clotting process. Okay? So we talked last term a little bit about blood thinners, and I said there were other blood thinners. It turns out that NSAIDs are very good blood thinners as well because they inhibit the production of prostaglandins that will favor production of platelets. Or favor the stickiness, not the production, but favor the stickiness of platelets. Okay? Thromboxanes in particular can be very important in this process. They can really help in that clotting process by making the platelets become sticky. Okay. There was a lot of interest. Gesundheit. That was emphatic. I like that. There was a lot of interest among drug companies a few years ago to find some very specific prostaglandin synthase or COX enzyme synthase inhibitors. And the reason was as follows. It turns out that we have at least 3 different sets of COX enzymes in our body, and there's probably more. All right? The COX-1 enzyme is pretty ubiquitous through our body, and it is inhibited by aspirin, as are the other COX enzymes. And so one of the places that problems arise in the taking of aspirin is due partly to the fact that the COX enzyme, COX-1, all right, makes a prostaglandin that stimulates the development of intestinal cells. So some people who take aspirin over a long period of time because they've got chronic pain find, oh, aspirin, man, it really nails my stomach. Well, why is it nailing their stomach? They're inhibiting the synthesis of prostaglandins that favor the development of intestinal tissue. And if they don't have that tissue developing as rapidly, what happens is you get ulcerated sores and so forth, problems where that tissue that turns over very rapidly is not being replaced as rapidly. So that's the root of problems associated with aspirin intake in some people. It doesn't happen in everybody. In some people with intestinal issues. Yes, Karen?
What about, I think it's called the
Enteric-coated aspirin? Yeah, trying to get it past that point where it's necessary. I don't know. That's obviously the strategy is they're trying to get that past the point where those prostaglandins are being made and needed. I don't know that. I don't know how effective that is, to be honest with you. Okay. The whole business of bufferin. Everybody's heard of bufferin? I mean, people used to run around and actually think that there was enough acid in aspirin. That's what caused the problem. You know? I mean, it's one of the stupider notions that you can have because if you think about HCl that's in your stomach, there's a heck of a lot more of that than there is in a little tiny aspirin you're taking. Right? Is your question or comment? You're shaking your head. You like the silly voices? A little helium.
That was pretty nice.
Okay.
My question was the prostaglandins of the GI tract that are affected that lead to these stomach discomforts, do they have something to do with controlling the production of new cells in the crypts inside the...
Yeah, I don't know. There may be, maybe other cells, or I don't know exactly the location of the cells, Jody. I don't know. Yeah. Okay. Now, I said that there was interest in developing specific inhibitors, and the specific inhibitors that people were trying to develop were to inhibit a different class of cyclooxygenases. These were known as COX-2 enzymes. And it turns out COX-2 enzymes were discovered because they appeared to be produced mainly in joints. People take aspirin for arthritic joints, for problems associated with that. And for the most part, the idea was if I can make a COX inhibitor that not inhibits COX-2 enzymes but not COX-1 enzymes, I can have my cake and eat it too. I don't have to worry about what's going to happen to my digestive system, and I can still relieve the pain associated with these. So billions of dollars went into the development of COX-2 enzymes. And they were released on the market, and it was a disaster. And you probably know some of the disaster. The disaster was that it turned out that people taking COX-2 enzymes, many of the COX-2 enzymes, found that there were heart problems resulting from that. And it turns out that the heart is another tissue where the COX-2 enzyme is being made. Okay? So most of the COX-2 enzymes got pulled off of the market. And that was a big debacle, as it were. Okay? Celebrex. I think Celebrex is still on the market. Anybody know about that? Yeah, Celebrex is one of the COX-2 inhibitors, and I think it's the only one that's still left on the market. Aleve and things like that, many of them got pulled because of concern over damage to heart tissue that were happening. And I don't know why Celebrex is still there. Maybe it actually doesn't have as much of an effect on the heart enzyme. I don't know. Okay. The last thing I'll say about this, oh yeah, question.
Do you know if during migraines, possibly these are produced?
Oh, that's a very good question. Do I know if prostaglandins are produced in migraines? If I were guessing, I would say that's not the major pain associated with migraines, but I would only be guessing at that. The pain of migraines is usually very intense and very different from the pains associated with regular headaches, and so I would wager that migraine headaches are more tied to neurons than it is to production of prostaglandins, but I don't know. It's a very good question. Okay. The last thing, oh yeah, 2 questions over here. Yeah, down here. The aspirin what? I'm not aware of aspirin being linked to blindness, so that's news to me. Yeah. What do you know? What the study was?
No, it was, I don't, but it was just like a more recent.
I haven't heard that. I haven't seen that. If you see it, I'd be curious to see the article. Lawrence?
Prostaglandin synthase and a type of COX enzyme, or is the COX enzyme a type of prostaglandin synthase?
Okay, so his question is prostaglandin synthase a type of cyclooxygenase, or is a cyclooxygenase a type of prostaglandin synthase? And they're synonymous terms, so they're equal. Yeah.
Are prostaglandins the ones that would trigger nerve cells then to tell your brain that there's pain in that area?
Yeah, so do prostaglandins trigger nerve cells to tell brain there's pain in that area? Probably not. Probably the pain that arises partly from the inflammation that they cause. So you feel pressure and so forth associated with those, and that's probably what's happening with it. Yes? How do prednisone do what? How does prednisone have so many effect on so many types of illnesses? That's a very good question. They're not all through this enzyme. So that would take more than I have time to answer here. But yeah, obviously there are other effects that steroids will have on us, which is why we want to avoid steroids in general. Okay, we're almost done, and I'm not going to finish early, but I will say one last thing here, and that is the pathway going up, the linear pathway. Okay? The linear pathway is important, and we're realizing increasingly the importance of the linear pathway going up because leukotrienes have been implicated in asthmatic attacks. Gesundheit. A little asthma attack went back there. All right? So there's a very active pathway right now, or a research process going on where people are trying to identify the role of leukotrienes and related compounds in triggering asthma attacks. And the idea being that if you can inhibit the production of leukotrienes, specifically by perhaps inhibiting lipoxygenases, you may have a very new and interesting effective treatment to prevent or reduce the severity of asthma attacks. So that's an important consideration as well. All right, I think that's a good place to stop. Let's stop there, and I will see you guys on Friday. How you doing, Liz?
Narcotics,
What's that?
like narcotic painkillers, do they?
No,
act on that same
no.
pathway? Okay.
Narcotic painkillers are acting to deaden the centers in the brain.
That's what I was thinking.
Yeah,
I didn't.
yeah.
Thank you.
Yep, you bet. So if you find that article, I'd be curious to see it. You got it? Okay.
Yeah, it's macular degeneration. Later, right?
Oh, okay.
Well, I got all Saturday. It was like really
Huh. I hadn't heard this. Interesting. I don't know why that would be, but you might suspect maybe a prostaglandin link. Yeah. Okay.
So for the
Come on back
midterm-
because I'm doing this.
Okay.
Yeah. What's up?
So for the midterm, I studied.
Oh my goodness. Dropping money. Thank you.
I studied quite a bit, and I felt like I had a good handle on the material. But
Uh-huh.
I wasn't expecting the questions that you had on there.
Okay.
So what does do to study more next time?
Well, I guess I would be curious to know what you, was it mainly that you just didn't study as many things as you needed to? Was it that you didn't understand what I was talking about?
I just did it.
Excuse me, I'm sorry.
I know you're.
Say what?
I guess I just did it broad. I studied the whole citric acid cycle and all the enzymes. I just didn't know the little subgroups or
Okay.
some of the